Thank you, ma'am. I wholeheartedly welcome all of you. I welcome Prashant sir, Sandeep sir, and all the Renuka ma'am and all the lovely students for today's introduction to bioinformatics sessions. So, in today's sessions, we will be talking about what is bioinformatics and what is the need of study. So, how bioinformatics deals with decoding biology of DNA, RNA, protein, which helps us in disease diagnosis, drug discoveries, in aiding our medical health facilities via various tools such as sequence alignment through BLAST, phylogenetic analysis, protein interaction, protein modeling, docking, molecular simulations. For now, I know these terms are very new to all of you, but by the end of this session and by the end of this complete 6-month course of BiXs, you will be very familiar with these terms and you will be very good to go and using these terms by your own. So, in the coming sessions, you will be learning more about statistics, data analysis methods, various programming languages, how we structure data, and what are databases. Databases, for starters, are the data house of information where we store specific information, and whenever required, we can retrieve back. In this way, this helps us to build a strong communication with scientific community. So let us start our today's session with the very foremost question: what is bioinformatics? Here I want to ask students Aradhya and Anaya, can you please recall what is bioinformatics from your last class? Can you please unmute your mic? Yeah, okay, okay. Thank you for your answer. Yeah, you are correct. Bioinformatics is a wholesome approach to solve biological problems. No matter how complicated your problem is, bioinformatics always comes with tools and software which helps us to understand better concepts of biology. So, in literal terms, it is a computational approach that we use to solve biological problems. It is an emerging and advanced branch of science that contains that mix biology with mathematics, computer science, physics, chemistry, and every domain of science. This information technology is applied for the management and analysis of biological data to combat major issues like storage and analysis. It is a logical and technical approach by which we can not only solve the problems but also we can predict new aspects from it. See, this chart very well means depict how bioinformatics connect all the dots of science, from engineering to computer science, chemistry, biochemistry, biology, maths, and whatnot. So, what is the big question now? How do we start with bioinformatics? We start with the base of three datas. Um, you all must have studied about central dogma of biology. Let's start with the basic biology. As we all know, biology is the branch of science which deals with organisms and their life processes. So we know life processes starts with this DNA molecule. This DNA is a very specific entity that encodes the whole being of an organism, meaning every single organism on this earth have its individual specific DNA which transcribes into RNA and forms protein. This protein further functions all the life processes. So, as I told, this DNA is very compactly uh, built on this chromosome which is present in the nucleus of a cell. This DNA is a very long stretch of nucleotides where specific genes are present. These genes contains instructions for making proteins. Further, these proteins may be individually or in coordination with other molecules and proteins functions perform functions. So this is an example, like um, I'll quote an example to make this better. If we have, say it's my birthday today, and it's, what is the time now? It's 5:10. So I ate a lot of cake. I drank a lot of Coca-Cola, and I ate a lot of sweets. So right now my body is having a lot of blood sugar, right? So now my mind is triggered that I usually don't have this level of blood sugar, but since I have ate so much of sugar, my blood sugar levels are very high. So my brain would signal my pancreas to secrete insulin. What insulin does is, this will activate the genes that are specific for making up insulin. This insulin is a protein which combats the high blood sugar level and stores the extra glucose in the form of glycogen. So you understand now what is happening here. This is a normal biological process where my DNA transcribed RNA specific for the formation of insulin protein. So how we translate this information to our information technology, to our laptop, how this this computer will understand this process. So for this, we have a specific codes that we feed in our computers. See, this is the example of ISR1 gene, which is 68,508 base pair long gene that encodes a protein which helps in insulin production. So if there are any mutations in any of the, if there are any mutation in any of the nucleotides, this will cause a altered protein sequence. This will ultimately produce a altered protein. You understand? So this is the basic approach of bioinformatics, that we study genes, we study proteins, and we see what are the changes that are leading to altered form of protein that we want to study. So this here is a table. I think you all are in 10th standard and you must be familiar with these 4 nucleotides. When I talk about DNA, our DNA is made up of 4 nucleotides that are present in triplets. These are known as codons. These triplets has a specific organization to form a specific amino acid. These amino acids uh, come in a long sequence to form protein. These amino acids, when come in a specific sequence, they form 3D structure, and this 3D structure then forms the functional protein. See, this is a picture of a building block that we used to play in our childhood. See, assume if these 3 dots are nucleotides, these 3 dots form such a structure of amino acid that it is very specific, and it combines with another specific amino acid of different composition to form the final overall structure. This is how nucleotides are combined to form amino acid, and amino acid forms the overall protein. Hope this cartoon makes it more clear. So, in the coming classes, in the next class, you will be learning about molecules and proteins. You will be learning about what are proteins and how their structures are formed. As I was telling you, see, I was talking about protein here. So this sequence is a long stretch of amino acids in a specific pattern, which is specific for this structure. So this here is a primary structure, and this here is a tertiary structure. So in the coming classes, you will learn in detail how proteins are formed, how their structures are maintained, how these proteins are derived and stored in data banks, and these data banks helps us to retrieve data whenever we want to study further. In the next module, in the third module, we'll be learning about microbiology and its relation, how we better interpret microbiology with the help of bioinformatics. We all know what are bacteria. I think we know. Can anybody answer me what are bacteria? What do you understand by bacteria? Okay, okay, I'll tell you. Uh, G, very good. Yeah, correct. So for a very basic idea, for now I'll differentiate bacteria into good bacteria and bad bacteria. We know what are good bacteria. We have heard about probiotic drinks. We know curd has bacteria in it. So bioinformatics helps us to identify such bacteria with the help of phylogenetic approaches. Phylogenetic approaches are to classify these bacteria based on their origin and evolution, how these organisms has evolved in the course of time, in the course of evolution. So phylogenetic analysis helps us to classify organisms on the basis of their evolution. Okay, this is an example where we can classify bacteria. We'll learn more about microbiology in our third module. Now coming to the fourth module, where we will learn about genetics. Genetics is a branch of science which deals with all the genetic material that we inherit. Right? As I told you, every single cell has its own set of complete genome, right? But it depends which cell will translate what protein. It is very specific. Like insulin insulin gene is present in every cell of our body, but it is only translated to protein in pancreas, right? So this way we will learn more about genetics. In our fifth module, we will be learning about protein interactions and system biology. So far, we know we have information in terms of genes, we have information in terms of molecular structure, in terms of proteins. So, and we also know we have different biochemical functions as well. Means we have different metabolites that are also produced in, along with proteins that helps in the proper functioning of phenotype. Means earlier we used to study genomics, which is the science of the branch of the techniques for gene studies. We had proteomics and we have metabolomics, read and studied differently, but later we realized that it is the overall system which needs to be studied. It is not the individual genes which affect the phenotype, but it is the combination of all processes that gives out the final output. So in system biology, we will learn how these different domains of science interact with each other, different domains of biology interact with each other to give an overall system of biology. And we will learn how these bioinformatic information helps us in novel drug discoveries, in various disease diagnosis, in understanding the metabolism behind normal functioning and the diseased functioning. And this way it helps us to increase our information of understanding disease and their progression. So we have various tools that aids in finding out how the diseases can be diagnosed, how the drugs can be delivered so that they are very efficient and has the minimal side effect. These days gene therapy is a very popular therapeutics that is used above drugs. We'll understand about those areas as well in the coming classes. So in this example, since we know now we have acquired a good information regarding metabolomics and genomic studies, this enzyme replacement therapy has recently provided treatments for diseases that were previously untreatable and had no response for drugs. So this way, bioinformatics is helping us to uncover various disease and their progressions. In the coming modules, we'll be learning about statistics. Statistics is more of the structured teaches you how to plan and structure your study, how to contribute different attributes, how to use different attributes, and how to perform our experiments so as to give the final inference. And in last, we'll be studying about big data analysis, big data science, where we'll be learning more about what is data, how we should collect them, and how we classify them for further analysis. In this, we use computer approaches, we use algorithms, machine learning approaches to predict things that we cannot see by just raw data. So we classify them and we apply uh, various algorithms to bring out various results. I'll tell you an example for this. Mm, in microbiology, I told you that we use phylogenetic analysis to to specify what bacteria it is. So for phylogenetic analysis, we need to have genomic sequence. While I was stating the gene for insulin, same way we need to extract that genomic information for say we want to just characterize, we want to just see what are the bacteria that are present in our surrounding, in my city. So what I will do, I will collect the samples from various sources for bacteria analysis, and the first approach is I'll go and retrieve their genetic information, their gene sequences, and then I'll do the phylogenetic analysis to discover that okay, this is a good gene, oh, this is a good bacteria, this is a bad bacteria. This is one approach. And another approach is that I take the sample, I isolate a single organism, and I take a picture. I take a microscopic picture, okay, and then I build up a machine learning, artificial intelligence based software where I just click my microscopic picture and feed in that software, and that software will use all the data of that particular picture from databases, their gene sequences, maybe their protein sequences, what are the metabolites that are produced in its life process. It will take all the data from databases and will tell you, okay, the picture you have given me was this bacteria, this was the name of this bacteria. So this process also saves a lot of time and money. So this is also one approach. um, One common example for this is mm, Google Lens. You just take a picture of a random insect that you find in your place in your room, and you just feed in the Google Lens. It will automatically tell you, okay, this insect that you have given me is this. Its name is this, its classification is this, and it is usually found. It will give you all the information that it has. So likewise, in similar phenomena, we can create various algorithms and various softwares which can help to save our time and money. Um, Thank you. That's all for today. I'll love to have your questions. Aradhya, Anaya. Okay, can I please request you to open up your webcams and mics so that we can have a good interactive session? Yeah, thank you, Radha. So, did you understand today's concept? Okay. Do you have any queries? One second. Sorry, Radha, I couldn't get your question. Can you please repeat? Yeah, sure. By the way, Aradhya, in which class are you? Yeah, so you must have learned about cell DNA by now. You must be familiar with the structures and functions of DNA. Okay, then it will be better for you to understand. See, what earlier we used to think that, say, a gene that I showed with an example, this gene IRS1, insulin receptor substrate 1. Okay, so I used to think earlier, when I was only familiar with DNA, that if I change any of the letter, any of the code here, it is a repetitive code of A, T, G, C. If any change occurs here, this will automatically give rise to a defaulter insulin. है ना? That insulin might have a different function that it should have had before. But this is not the case. Sometimes there are various check mechanisms as well. If there is any if there is any default in transcription of RNA or during the translation of protein, our cell also have various mechanisms that can uh, track that default and that can redo it. Okay, maybe it will not let that protein to get a final functional state. Okay, so our cell has this capability. So what does this conclude? That a single mutation in the DNA is not wholly responsible for the mutation in the whole protein. Maybe our cell will rectify it. Okay. Cancer is one such example where our body have various checkpoints where it check any defaults in transcription and translation. Okay. So by saying that, it is clear that only genomics is not responsible for the phenotype of body. Phenotype means for my whole body to function. The genomics is not wholly responsible for the production of maybe diabetes. Okay. Same way, proteomics also have post translation modifications where the default proteins are taken out of the system. Same way, we cannot study individual जैसे metabolites. This is a function of whole three domains together to form system biology. Not one system is responsible for the phenotype. All the three systems in coordination with each other then give rise to system biology. So you'll study more in detail about system biology in the coming classes. Thank you. Are there any other questions? Uh, Sandeep sir will be taking your next session for molecules and proteins. Renuka ma'am, you want to add something to this? Thank you, ma'am. I think Anaya left the meeting. She had network issues. Yeah, 1 minute, ma'am. I'll stop sharing the screen. Yeah, one, two, three. Smile. Yeah, ma'am, you can also take. Yes, ma'am. Thank you, ma'am. Thank you, Sandeep, sir.